Welcome, everyone, to our lecture on the comparative anatomy of vertebrates. Today, we are diving into a truly fascinating topic, the classification of receptors. Have you ever wondered how a shark hunts in murky water, or how you instantly pull your hand away from a hot stove? The answer lies in specialized biological sensors. In this video, we will build your understanding of these receptors from the ground up. We will explore what they are, how they work mathematically, and how we classify them across the animal kingdom. Let us begin our journey into the sensory world. To start, let us imagine you are walking barefoot in a park and suddenly step on a sharp pebble. Instantly, you feel pain. But you also feel the warm sun and hear birds chirping. How does your brain process all of this simultaneously? The secret lies in tiny, specialized biological sensors called receptors. Think of them as your body's frontline reporters. They are stationed everywhere, on your skin and deep inside your organs. Their primary job is to constantly listen to the environment. When a physical event occurs, like a change in pressure or light, these receptors translate that physical event into an electrical language. This is the only language your brain can actually understand, allowing you to perceive the world around you. Now that we understand their general role, let us look at how we formally define them in comparative anatomy. A sensory receptor is a specialized cell, or sometimes a modified nerve ending, designed to respond to a specific environmental stimulus. Crucially, these structures act as biological transducers. In physics, a transducer converts one form of energy into another. Here, the receptor converts physical energy, like mechanical pressure or light waves, into an electrical signal called a receptor potential. If this initial electrical spark is strong enough, it triggers a full action potential. This action potential then travels rapidly along sensory neurons directly to the central nervous system, delivering the message to the brain. Building on that definition, it is important to know that receptors are highly specialized. This brings us to the law of specific nerve energies, which states that a receptor designed to detect light will absolutely never respond to sound waves. Why does this strict specificity matter in the real world? Because without it, an animal would be completely blind, deaf, and numb, making survival impossible. For example, sharks rely on highly specialized electroreceptors to detect the faint electrical signals of hidden prey in murky oceans, a sense we humans completely lack. In human medicine, understanding pain receptors, known as nociceptors, is what allows doctors to develop targeted anesthesias for life-saving surgeries. Receptors are the ultimate survival toolkit. Having seen why receptors are vital for survival, we must now ask, how sensitive are they? To truly understand how receptors work, we have to look at how they detect changes in the environment. Receptors do not just passively detect the presence of a stimulus. Their main job is to detect changes in its intensity. Think about holding a heavy rock versus a light feather. If you add a tiny pebble to the heavy rock, you probably won't feel the difference. But add it to the feather, and you will. This means the larger or stronger the initial stimulus, the bigger the change must be for your receptors to actually notice it. This fundamental principle of sensory physiology is described mathematically by Weber's law. This brings us to the mathematical heart of receptor sensitivity, Weber's law. The formula is written as delta I divided by I equals K. Let us break down exactly what each symbol means. The letter I represents the initial intensity of the stimulus, like the starting weight of an object or the initial brightness of a room. Delta I represents the just noticeable difference. This is the absolute smallest change in stimulus intensity that the receptor can physically detect. Finally, K is Weber's fraction. This is a constant value that is unique to each specific type of receptor. Physically, this equation tells us that if the initial intensity I increases, the required change, delta I, must also increase proportionally to keep the fraction K constant. Now that we have our formula, let us see this with a concrete example. Imagine a vertebrate's mechanoreceptors, the pressure sensors in its skin, have a Weber fraction of 0.05 for detecting weight. The animal is holding a rock that weighs 100 grams. We want to know the absolute minimum additional weight required for the receptors to fire a new signal. We are given I equals 100 and K equals 0.05. We need to find delta I. By multiplying both sides of Weber's law by I, we rearrange the formula to delta I equals I times K. Substituting our numbers, we get 100 multiplied by 0.05. The final answer is 5 grams. Physically, this means if you only added 2 grams, the animal would not feel any difference at all. Let us reinforce this with a second example, this time looking at vision. Consider a nocturnal vertebrate's photoreceptors. These are light-detecting cells in the eye. They have a Weber fraction of 0.08. The ambient light intensity in the forest is currently 50 photons per second. What is the minimum change in brightness the animal can detect? Again, we state what is given. I equals 50 and K equals 0.08. We use our rearranged formula, delta I equals I times K, because it easily transforms a ratio into a straightforward multiplication problem. Plugging in our numbers, we multiply 50 by 0.08. The final answer is 4 photons per second. This means the light must increase to 54 or drop to 46 for the animal to notice, highlighting how sensitive nocturnal eyes truly are. Now that we understand how receptors detect changes mathematically, we must classify them. The first major way to classify receptors is based on where they are located in the vertebrate body. There are 3 main categories here. First are exteroceptors. These sit near the surface of the body and monitor the outside world, handling touch, vision, and hearing. Second are interoceptors, also known as visceroceptors. These are hidden deep inside your internal organs and blood vessels. They quietly monitor things like blood pressure and oxygen levels, usually completely below your conscious awareness. Finally, we have proprioceptors. These are specialized sensors located in your muscles, tendons, and joints. They constantly map your body's position in space, which is exactly what allows a cat to land on its feet, or you to touch your nose with your eyes closed. Besides location, the most common way to classify receptors in comparative anatomy is by the specific type of energy they detect. Let us look at the first 3 types. Mechanoreceptors respond to physical deformation. Whenever there is pressure, stretching, or vibration, these receptors fire. They are responsible for your sense of touch and even your hearing, thanks to tiny hair cells in your ear. Next are thermoreceptors. As the name suggests, these are dedicated to detecting changes in temperature. Interestingly, vertebrates have completely separate, distinct receptors for hot and cold stimuli to protect the body from thermal damage. Third are photoreceptors. These are highly specialized cells located in the retina of the eye. They contain unique pigments capable of absorbing light energy and converting it into electrical signals for the brain. Continuing with our classification by stimulus type, we have 2 more critical categories. Chemoreceptors are designed to detect specific chemical molecules. When you smell a flower or taste your food, you are relying on exteroceptive chemoreceptors, but they also work internally, monitoring vital blood chemistry like carbon dioxide levels to regulate your breathing. Finally, we have nociceptors. These are the body's dedicated pain receptors. Unlike other receptors that detect normal changes, nociceptors only fire when they detect extreme, potentially dangerous stimuli. This could be extreme mechanical pressure, burning heat, or toxic chemicals. While pain is unpleasant, nociceptors are absolutely essential. Their primary function is to act as an alarm system, protecting the animal from severe injury and ensuring its survival. Let us bring all of this classification together visually. What you are looking at is a comprehensive flowchart that maps out the entire sensory receptor system we just discussed. At the very top, we have our main subject, sensory receptors. Notice how the chart splits into two distinct branches. On the left, we classify them by location, breaking down into exteroceptors for the outside world, interoceptors for the internal organs, and proprioceptors for body position. On the right side, we classify them by stimulus type. Here, you see the 5 specific energy detectors: mechanoreceptors for force, thermoreceptors for temperature, photoreceptors for light, chemoreceptors for chemicals, and nociceptors for pain. This diagram perfectly illustrates how a single receptor can be classified in 2 different ways simultaneously. We have covered a lot of ground today, so let us summarize the most important ideas from this topic. First, remember that sensory receptors are vital biological transducers. They are the only way a vertebrate's nervous system can connect to the outside world and monitor its internal state. Second, we learned that receptor sensitivity is not random. It is governed by mathematical principles like Weber's law, which dictates how big a change must be to be noticed. Third, we can map out an animal's sensory capabilities by classifying receptors based on their location and the specific type of stimulus they detect. By understanding these classifications, we gain a profound appreciation for evolutionary biology. You now understand exactly how vertebrates perceive and survive in their incredibly diverse habitats. Great job today!